Now for something completely different, but a tiny bit of housekeeping beforehand. I've been asked to let everybody know we have this session, we have some lightning talks, and then after that, there's going to be food, beer, and wine. So get comfortable. You've got 1 hour to go. And it is my great pleasure to introduce the guy standing behind me, Vaughn Cooper, who is a professor of evolutionary biology. And he's going to give you a talk about something that really shut me down when I first read this report. And I'm going to stop right there. Go for it, Vaughn.
Thanks, John. It's a huge pleasure to be here at this meeting. I had hoped to attend the previous iteration where my sort of home society, ASM, was affiliated, but I'm delighted to be here, and thanks for the welcome. It's been great. So I'm used to being the person giving the kind of outsider talk as an evolutionary biologist at a medical school, but this may stop you in your tracks. Okay, so I'm here to speak about the prospect of mirror life. And so, yeah, I should say I'm president-elect of ASM. So if you want to talk to me about ASM stuff afterwards, please do. And so just as an outline, some background of how we got to this topic. We'll talk about what mirror life might be, its feasibility and any potential benefits, its risks, which I'll point out are substantial. Talk about containment and countermeasures. And some summary work from a great deal of work that's been ongoing over the last 18 months, along with some recent progress. So some background, and I should note that I'm going to go relatively quickly because I do want time for Q&A at the end. So back in the 1840s, Pasteur was, many of you may know, was not only a great scientist, but also an artist, and had been literally drawing and making lithographs and studying mirror images of his own art and thinking about that. And he brought that mindset as he was looking at crystals produced from heating up wine too much and then studying the crystals that were being deposited on wine vats, and discovered the nature of chirality. I mean, the notion that there were sort of different forms of crystals that would refract had been noted earlier, but it was Pasteur who sort of identified that molecular structure, and then quickly from that began to think and notice that all life on this planet is homochiral, right? We all have nucleotides and proteins produced by the ribosome of the L form. But that made him wonder, and others wonder, whether life might have arisen in the mirror. And so if you're thinking about what the simplest form of mirror life would be, it would be a mirror microbe like E. coli. And that's basically what I'll be talking about today with that notion in mind, a mirrored E. coli. And so, and with that, it's also important to know that what that would mean is that all of the proteins produced by this mirror cell, if it ever existed, would be just like the proteins made in our current body or in the E. coli's body, and it's completely in the inverse. So the folding would just be inverted, the same sequence, but made of the opposite form molecules would fold in the opposite. And so this idea had been percolating among science fiction. But in 1992, there was a letter by a couple of authors who stated we need to have that mirror life would have built-in immunity to attack from normal life, and that prospective synthesizers of life would need to consider these matters in detail before getting started. Fast forward about 2 decades, there's an article in Wired stating that mirror life wouldn't have any predators or diseases to limit its reproduction. The synthesizers would have to keep an eye on that. And then a couple of years later, George Church and his co-author Regis discuss mirror life in another article. So this idea has been out there, but it was sort of foreign to me, other than the typical what I learned from organic chemistry in undergrad work, like many of you. when a couple of scientists came to me and said, in a secure location, would you take a look at this white paper in which we present this hypothesis that states that the synthesis of a mirror cell, which is becoming technologically more possible, poses a degree of threat that perhaps we've never thought about before. And so, like good scientists, they said, here, take a look at this hypothesis, do your very best to kill the hypothesis. Please falsify it. But if you can't falsify it, help us find other people who might be able to. So as an evolutionary biologist and ecologist, I focused hard on what I thought whether this mirror cell would do in nature or in interactions with other organisms other than Homo sapiens. And I agreed this is a problem. And then I had a hand in helping to build the team that took a look at it. And so just as an AI-generated cartoon, we put together a really diverse group of researchers over the coming months. This really started, by the way, in January of 2024. So this has been progressing quite rapidly. But by May or June of 2024, we'd assembled a team to produce, I should just slip to a team of authors from 5 continents, from various countries, to take a look at this idea, to weigh in, to offer feedback and commentary, and ultimately to come to some consensus that we published in this article, the Science Policy Forum paper that many of you may have seen, entitled Confronting Risks of Mirror Life, along with a really detailed, and I will say, self-serving, a very interesting 300-page technical report that was peer-reviewed by each chapter separately by subject matter experts, entitled Technical Report on Mirror Bacteria Feasibility and Risks. You will have technical questions. I will not be able to answer all of them. They are in this report in most likelihood, or we simply don't know. That led to a great deal of coverage, including an excellent piece by Carl Zimmer at the left there, entitling this The Prospect of a Second Tree of Life, as well as other noticeable publications. Okay, and so in summary, we concluded mirror bacteria could plausibly be created by life scientists in the next 10 to 30 years. They would have little practical benefit, although they would be extremely cool from those who are interested in synthetic life and astrobiology. That sort of wow factor is simply not worth the risk. And these bacteria, if created and released, could plausibly pose unprecedented risk to humans, animals, plants, and really all ecosystems. To manage this, global discussion and governance are needed, and we set out in that article a plan to do so, and I'll tell you a little bit about what we've been doing since the publication. And then our initial conclusion is that mirror bacteria should not be created, and that key enabling work should not be pursued or funded. So that's the top line point for this entire talk. Let me break it down a little bit. So how did we get here? So that is really, how did the researchers who presented the initial white paper come to me when they did? And it's really 2 things. First, the sort of overall synthesis of ordinary life, although that has not been done, right? No cell has been built from the ground up or from the top down yet. But that progress is accelerating. And it's accelerating because of a range of technologies involving the synthesis of very long molecules that you can stitch together, as well as progress on cell-free systems where you can build components of cells and build them up piecemeal. In addition, artificial intelligence is enabling more rapid iteration and feedback of some of these technologies. We currently know no researchers who are actively pursuing mirror bacteria. I do want to point out something really key about our author list. Several of our co-authors were people who had stated in grant proposals that they might pursue this down the road as a long stream goal of some of their work. Each of those people are now now have agreed that they should terminate that work. And this is really an amazing, I would say, selfless act and an example of leadership for the public good. However, progress in constructing mirror nucleic acids by some of these labs and proteins, as well as longer mirror molecules like RNA polymerase, has been done. And this work is, I will say, incredibly cool, that you can actually build a mirror polymerase that will synthesize kilobase lengths of mirror ribonucleic acid. And those enabling technologies were the things that made us think this is actually technically feasible given enough time and effort. Those timelines are quite uncertain. It would take about 10 to 30 years sort of by default, but it really is more a matter of resources and motivation, right? So, and as I mentioned, AI could accelerate these timelines. And I'd say if we have a minimum time 10 years from today, if we do nothing and make a decision in 5 years' time, we won't have 10 years from 2030 because these technologies are moving inevitably. The good news is the foreseeable benefits of producing a mirror cell appear quite limited. The primary motivation is basically to help accelerate the exploration of mirror molecular space. Some of those molecules have shown promise as diagnostics, primarily because proteases don't work on opposite form peptides. And so you could imagine synthesizing a biologic that is particularly long-lived because your proteases are ineffective at breaking it down. Other reasons might be that you might synthesize a molecule that is relatively invisible to the immune system, and thus it won't have ancillary or sort of side effects of dosing. So some of these mirror biomolecules do show promise as therapeutics, but importantly, they can today be produced without a mirror microbe, and thus we really do encourage the development of alternative technologies to explore this space. You might be thinking, well, okay, you now synthesized this cell. What's the prospect of it actually surviving in nature? This was my initial stumbling block when I was looking at the sort of the prospect of synthetic life. But it was soon that we sort of went back to our initial, old microbiology literature and discovered that, E. coli can grow on a wide range of resources that are achiral. And those resources listed here are actually quite abundant, even at levels multiple orders of magnitude above by concentration in the body than are necessary for their growth. So for instance, in the gut lumen, many of these nutrients that are at left are present at micrograms per milliliter, whereas the estimated minimum required concentrations for E. coli to produce another E. coli de novo just from a sole carbon source is 1 or 2 order of magnitudes below. So they can grow. And you might also be thinking that this initial engineered cell will be incredibly fragile and deliberately handicapped. But it's important to recognize, that those sorts of barriers are easily overcome. And really the functioning model is an E. coli, not this fragile cell that really has never lived outside of in the world. So what makes mirror cells so dangerous, or mirror bacteria so dangerous? They are largely invisible to all predators, starting with the predators in your own body. So they will not be seen and detected by your phagocytic cells, and your adaptive immune system will fail. So here's an example, some mirror biomolecules. Nope, there we are. Mirror biomolecules could invade innate immunity, and it's particularly because proteins resist degradation, which impairs T cell responses. So these pattern recognitions of all of the arms of your immune system are sort of baked in and really evolutionarily ancient. And it's critical for innate immunity. And then further, it is the processing of pathogen-associated molecules involving degradation that is key for the adaptive immune responses by T cells. As an example of how this might look, mice that are deficient in TLR2 which would be one of the many mechanisms of pathogen recognition that would fail in this instance, die quite rapidly relative to immunocompetent mice. Likewise, at right here, we have an example of the rate of peptide cleavage by an enzyme, and that enzyme cannot cleave D peptides, shown here in dark. So this is the rate of cleaving of an L peptide and the rate of cleaving of a D peptide. And so consequently, that lack of cleaving means no antigen processing and no activation of T cells. And then likewise, impaired T cell activation would impair the production of antibodies. And as a great example of what happens when you can't produce effective antibodies, this is a fairly well-known, maybe a well-known example to many of you in the audience. Patients with impaired MHC2, impaired adaptive immunity from an inborn MHC2 deficiency, are extremely susceptible to infections. And those who have these deficiencies, even with bone marrow transplant, have very low survival, and without bone marrow transplant, live only up to 10 years. And so you can imagine this is just one deficit, and we are positing that would be a panoply of these deficits all mixed together. So that's us. We would be in trouble should we be accidentally infected by a synthesized mirror cell. What would happen if that person who is accidentally infected went out and became the vector to infect nature? We believe that natural chirality bacteria, mirrobacteria would be completely immune to their natural predators. And those are phage and amoeba. So there will be no phage that would actually be able to kill the mirror cell. And since phage are largely accountable for about half of their death every day, you could imagine, despite their initially slower growth rate in this foreign environment, with the dependency on achiral or opposite chiral nutrients, which are rarer, that very low growth rate means that they will inexorably expand in number, and they will do so in virtually all ecosystems, including plants and a range of animals. And you could also very easily imagine that dispersal worldwide facilitated by insects would happen rapidly within a matter of weeks or months. The potential result is massive ecological harm, extinctions, and degradation of habits. And I also want to point out that even though they do not actually have to outcompete native microbes, which of course live on a microbial planet, they would have a lot of competitors. They don't need to outcompete all of them, but they will persist because of the absence of predators. Containment is not going to work, we don't think, primarily because malicious actors could likely replicate methods and construct ways to make them more robust. And frankly, I don't wish to alarm any of us who work in biosecure facilities, but accidents do happen. And given the severity of the risk that we're positing, we want the risk of accident to be literally zero. Relevant to this audience, we believe that medical countermeasures can be developed and are worth pursuing, but they are not likely to be sufficient because, very bluntly, without sort of skipping over the details of this slide, you cannot you can't douse the world at once in effective antibiotics. So it is true that you can build sort of deform antibiotics that would be effective. You could easily engineer them. You can even engineer mirrobacteriophage, but they would only be effective against in that particular instance at the point source. And we know that resistance will evolve, and that will be a runaway process that you will never be able to keep up with. So our recommendations is that we need global conversation, and that's why I'm here, is to be part of this audience to facilitate that conversation. Our starting point for discussion is that mirror life should not be created, but mirror molecules for therapeutics and synthetic cells and broadly synthetic biology should continue. We then need really sort of, I would say, careful, thoughtful, sort of clever governance strategies to manage the production of precursor molecules that would be key enabling steps towards mirror life. And that research to better understand and prepare for risks is key. I just want to quickly summarize where we've gone. There was a terrific meeting in Paris and another one most recently in Manchester that I'll talk about next. But some immediate deliverables are remarks from UNESCO, which suggest that they impose precautions on mirror replicating cells, specifically enact precautionary global moratorium on creating mirror cells. And then the UK government said there should be a coalition among funders, researchers, governments, and society to prevent the development of replicating mirror organisms. Now, just really quickly, this meeting at Institut Pasteur was a first major kickoff moment that had 5 discussion tracks. I happened to co-chair the policy interface. I really encourage you to learn more about what we've done so far by following that QR code or going to parismirrorlife.org. And then very recently, a lot of the key stakeholders got together in Manchester to talk about really where are we? Where is the technology today? And they concluded importantly that the latest technology of synthetic biology poses no risks towards these aims of mirror life, but there was some healthy dialogue about where sort of the fire breaks in technology development might be instituted. That report is forthcoming. Those 4 precursor technologies were protein synthesis using recombinant elements, mirror ribosomes, which many of us believe is a red line, crossover translation systems, and then synthetic natural chirality cells. Okay, so I will just leave this to say this is where we're going, and I'll leave it there for discussion. Thanks very much.
So I hope you get it now why I thought this was a really important talk. And I'm going to ask a couple of questions fairly quickly, and then we'll take questions from the audience. What is the most advanced stage of creating normal chirality life? Can someone today make an E. coli from scratch?
No, they can't. So currently, no, that has not happened.
But how close are we?
How close are we? So we can synthesize essentially multi-megabase genomes and replace real cells, sort of normal cells with synthesized genomes. It's the synthesis of all the other parts that go along with it that match the synthesized genome, this sort of booting up a cell. Those technologies haven't been resolved. And what I just said is incredibly inefficient. So you can synthesize mirror genomes, you can synthesize real genomes, normal genomes from scratch, but the production of the cell is still a work in progress.
Yeah, I got to go to the Paris meeting, and the analogy I heard there was it's kind of like you need to assemble a car going 30 miles an hour down the road. All the parts have to pop into place, kaboom. And then
it's a perfect metaphor.
The car keeps going. Why do we only have one chirality?
We think it was literally I wouldn't say a frozen accident, but it's a self-reinforcing process that started early in really in the chemical origins of life that for some reason got a numerical advantage and then locked the rest of evolving life into that format, much like sort of VHS over Betamax for those of you who have a certain age.
So is there if we had locked into the other chirality, is there any reason to believe other than I would be right, I'd be left-handed. Is there anything in all your digging around that suggests there's a reason that the other format doesn't work?
There's no reason. No, so the answer is no. There are some, as you know, a few interesting hypotheses about parts of reactions that might be biased towards the format that we have on this planet. But I will note that when an asteroid was recently sort of bioprospected recently, they found equimolar flavors of both hands of organic molecules on that asteroid.
Fascinating. But I mentioned there's an AMR.Solutions newsletter where I learned a lot about this. And some of the things he is saying, he's taught me, and they're in the newsletter. So if you want to find a bunch of links, go to the AMR Solutions and look for the word mirror. You're going to find that. What's been the reaction to this at scientific levels, and is there precedent for basically saying, stop?
So I'd say the reaction has been startled, like you, critical about some of these key feasibility steps. Often they won't be able to eat, they won't be able to survive in the natural environment. We're pretty confident about that. That's not true because bacteria just do survive in nature when intentionally or unintentionally released. There's been some guardedness and some, I would say, overreaction aiming to persecute synthetic biology. And this is a reaction that we really wish to avoid and manage because immense, potentially life-saving solutions are going to come from that.
So you say solutions from, well, we actually use...
Well, here, right? Solutions here. But
right.
the accelerated production of novel antimicrobials that require the kind of chemistry that you can't really easily do outside of a cell.
Right. And clearly we take advantage of chiral molecules all the time in various things. And so it's not saying that chirality is making the L or the D version of something is inherently wrong. Okay, so I've gone through a couple of questions right there. So the microphone here to the gentleman with the blue tie. And I guess I need to get some microphones here. You take...
Me? No. Hello. Yeah, now it works. Strange. Dinesh Suarez from ASM, actually. So fascinating talk, Vaughn. I'm just curious if you've sought input or if your group included experts in protein structure. The reason I mention this is because at least in silico, from what I imagine, you could quite easily reverse the chirality just by undertaking transformations of the coordinates, of the three-dimensional coordinates that we already have. So a tool like AlphaFold could potentially be trained in a certain way with some cleanup of stereochemistry to actually give you the enantiomer, the mirror image. So I'm just curious, what are the thoughts if these experts have been included in the conversation, and what their thoughts and concerns are, and how can we actually leverage that knowledge?
Yeah, so the short answer is yes. And I didn't sort of land the key point. The thing to be celebrated about this initiative was that group of authors, many of them are luminaries with expertise in areas just like the one that you mentioned, were each independently presented this challenge. Each of them came to, over time, this consensus that we all agreed upon. That happened very quickly with very structured, focused, moderated discussion. And we were across time zones and continents and backgrounds and interests, and it included people who are in the policy space as well as people in the very basic physical sciences space. And we came together that quickly. And I think this is a model of how, I would say, big challenges can be addressed with a focused, and I will say, philanthropically backed effort in a hurry. And so, there are plenty of other global challenges, including AMR, that might be supported by this kind of initiative, including folks in this room.
So we're going to try one more question, Dan. It needs to be a quick question.
It'll be a quick one. So Vaughn, nice talk. That urgency that you're talking about in your asteroid thing brought a thing to mind. It's not just doing it here. We shouldn't make these things here. It's very obvious, right? We probably shouldn't bring them home either.
That's absolutely right.
From Mars. So I'm wondering, we know there may be evidence of life on Mars now in that wet area, and there's talk of NASA now bringing samples back. Maybe we shouldn't if they're right-handed.
Yeah.
And I'm serious about this. You bring that back, there's no containment. You already mentioned it with the asteroid. Could get everywhere. So maybe we leave that on Mars, let Elon go do that.
Right? No, you're absolutely right. And I feel this is like this unmentioned subtext is that first contact might be mutually destructive. Yeah, NASA's aware of this. We don't know if, I think there's still a lot. Let me just contextualize. We are more certain that mirror cells would pose these risks than I think the field is about life on Mars currently, but these precautions actually are baked in to NASA's sort of bioprospecting plans. Yep.
And with that, so Vaughn will be here this evening. You're going to need the beer and the wine, okay? All right. And so thank you for making the trip to be here.